welcome back to this video. Let us get started with a positive frame of mind. In order to create that, listen to this inspirational quote. If you quit once, it becomes a habit. Don't quit. Okay, the next unit is biotechnology. There are two chapters coming under this unit: principles and process of biotechnology and the applications of biotechnology. So we can study the first lesson, that is principles and process of biotechnology. The topics to be covered in this session are what is biotechnology, core techniques of biotechnology, aims of biotechnology, and the basic steps used for recombinant DNA technology, or the basic steps in genetically modified organisms. Biotechnology, the branch of modern biology, changed our daily life as its products brought qualitative improvement in health and food production. The basic principles underlying biotechnological processes and some applications are highlighted and discussed in this chapter. So, what is biotechnology? It is the techniques of using live organisms or their enzymes for products and processes useful to humans. The definition given by European Federation of Biotechnology, or EFB, defines biotechnology as the integration of natural science and organisms, cells, parts thereof, and molecular analogues for products and services. What do you mean by biotechnology from this definition? See, see this definition. Biotechnology is the techniques of using live organisms or their enzymes for products and processes useful to humans. Can you give one example? Example of old biotechnology: formation of curd from milk. Yes, in this, for the change of the milk into curd, there is an influence of the living organism named bacteria. The name of the bacteria is lactobacillus. That bacteria is responsible for the change of curd from milk. Then, what about the modern? That is one example. Can you say other examples? Formation of batter by using yeast. There are lots of examples for this. Can you give example for modern biotechnology? What all things are produced recently? Production of insulin, BT, brinjal, etc. are the examples. Next, what is the aim of biotechnology? Or biotechnology technology deals with what all things? In which all area the applications of biotechnology we can see? First one, microbe mediated processes that we have discussed, making curd, bread, wine, etc. Second one, in vitro fertilization. It is otherwise known as test tube baby. That program is known as in vitro fertilization. Next is synthesis and using of a gene. Synthesize new gene. Preparation of DNA vaccine. Correcting a defective gene. So biotechnology deals with all these things. What are the two important core techniques of modern biotechnology? They are, first one is genetic engineering and the second one is bioprocess engineering. Here, what is the difference between these two? What is genetic engineering? Yes, in this technique, genetic material is, that is either RNA or DNA, that is chemically altered and introduced into host organism to change the phenotype. The technique in which genetic material is chemically altered and introduced into host organisms to change the phenotype. Then what about bioprocess engineering? That is the maintenance of sterile ambience in chemical engineering process for growing desired microbe or eukaryotic cell in large quantities for the manufacture of antibiotics, vaccines, enzymes, etc. Then, what are the important steps in genetically modifying an organism? So, actually, what are the important steps of biotechnology? Recombinant DNA technology or genetic engineering. So, basic steps in genetically modifying an organism. First one is identification of DNA with the desirable genes. In order to understand all these things, you should go through another illustration, that is, look here. Yes, the important steps in order to create a genetically modified organism includes isolation of the DNA, insertion of this desired DNA in the plasmid vector, that is known as the recombinant DNA. Then again it is inserted into a host organism. It includes many steps. Then can you give one example for genetically modified organism? Yes, that is Bt brinjal. So here Bt brinjal, here Bt stands for Bacillus thuringiensis. So this brinjal is modified genetically. In order to modify this brinjal genetically, the desired DNA is taken from a bacteria. The name of the bacteria is Bacillus thuringiensis. All these things you will study in detail later. But this diagrammatic sketch will help you to understand how this genetically modified organism is created. It gives you an outline. The detailed study is there that we can see later. Then, whatever is the gene of interest, suppose pest resistant plant we want to create, you imagine that gene we have to isolate. That is known as gene of interest. Then, how do we um, isolate this gene of interest? That is enzymatically cleave DNA into fragments. So, these isolated fragments, these are the isolated fragments. Then, what is the next step? Directly we are unable to inject this isolated desired DNA into a brinjal plant. Instead of that, we use a vector. From where we get this vector? This is also from bacteria because bacteria consists of two DNA. One is chromosomal DNA. This is the chromosomal DNA. This is the chromosomal DNA and this is the plasmid DNA. So this chromosomal DNA controls the life activities of this bacteria. Then what is this plasmid DNA? Plasmid DNA is an extra chromosomal DNA. They are able to replicate themselves. So we have to select this plasmid as the vector. First, we have isolated the gene of interest. You imagine a pest resistant gene we are isolating, that is isolating by using enzymes. That isolated DNA, this is known as the isolated gene of interest. Imagine this is the isolated gene of interest. This one we have to insert into the vector. So where this vector is present, what is the name of this vector? Name of the vector is plasmid. And where is it present? It is present inside the bacterial cell. What is a plasmid? Plasmid is an extra chromosomal DNA that able to replicate themselves. So we have to isolate the plasmid. Then in the next step, insert this gene of interest, insert this gene of interest into the plasmid. There also we want to make a cut by using enzymes. After that, by using another enzyme, ligate it. Ligate means paste it. Okay, then what is this plasmid known as? This is known as recombinant DNA. Now its name is recombinant DNA. Once the desired DNA inserted into the plasmid, that is known as recombinant DNA. What is it called? Recombinant DNA. I'll write here. Recombinant DNA. Okay. Then recombinant DNA is inserted into the gene of bacterium, or it is inserted into the bacterium. Actually, that is the host. Allow them to multiplicate. Allow them to multiply. So actually, this is the outline of recombinant DNA technology. But this process is an elaborate, tedious, so that we have to study one by one. Okay, we can see basic steps in genetically modifying an organism. The three important steps are identification of DNA with the desirable genes. In the case of BT brinjal is the product. Then in that case, pest resistant gene is the desirable gene. Second one is introduction of the identified DNA into the host. Then third one, maintenance of introduced DNA in the host and transfer of the DNA to its progeny. Okay. In the second step, you can see introduction of the identified DNA into the host. So, in order to do this process, we require many tools like vector, plasmid, enzymes, etc. In the case of identification of DNA with the desirable gene, Only the desirable genes are introduced in biotechnology or recombinant DNA technology or genetically modify organism. Why? Because traditional hybridization, if we use traditional hybridization, lead to inclusion and multiplication of undesirable genes and desirable genes. In order to avoid this situation, in genetic engineering, we have to isolate the desirable gene only. So that is the meaning of the first step, identification of DNA with the desirable gene. Then second step, introduction of the identified DNA into the host. In order to do this particular process, a vector DNA such as plasmid is used to deliver an aligned piece of DNA into the host organism. So what is the meaning of this aligned piece of DNA? So this aligned piece of DNA, that means gene of interest, desired DNA, target DNA, etc. are the various names. So write down these names. What are they? Aligned piece of DNA means desired DNA, desired DNA. Then what is the other one? Target DNA. Then aligned piece of DNA. PIECE, piece of DNA. Then gene of interest. So these are the various names we have used in the desired DNA. So here one spelling mistake, please correct it. Desired DNA. Desired DNA, target DNA, aligned piece of DNA, gene of interest. So whatever the gene we require in order to get a recombinant DNA, that is known as desired DNA. Okay, then plasmid, it is known as the vector that is also present inside the bacterial cell that is used for this process. See, bacterial DNA, plasmid DNA, recombinant plasmid. That means once the desired DNA is inserted, that is known as recombinant plasmid or recombinant DNA, then it is introduced into the host cell. It is known as the transformed cell. That is the second step. Then what about the third step? Or what is the third step? Maintenance of introduced DNA in the host and transfer of the DNA to its progeny. Now it is received by the host. After that, that expression we should see in the progeny. So that is the third step. Maintenance of introduced DNA in the host and transfer of the DNA to its progeny. A piece of aligned DNA has now the sequence called origin of replication. Origin of replication means the place where the replication start, which is otherwise known as ORI, origin of replication. A piece of aligned DNA has now the sequence called ORI that is needed for starting replication. So, it cannot multiply itself in the progeny cells of the organism. Hence, aligned DNA is integrated into the recipient genome. In the recipient genome, ORI is there. Origin of replication is there. It multiplies and inherits along with the host DNA. So these are the important three steps of recombinant DNA technology or basic steps in genetically modifying an organism. Which are they? First one is identification of DNA with desirable gene. Second one, introduction of the identified DNA into the host. And the third one, maintenance of introduced DNA in the host and transfer of the DNA to its progeny. You can see the name of two scientists. They are Stanley Cohen and Herbert Boyer. They produced the first recombinant DNA in 1972. First recombinant DNA was produced by Stanley Cohen and Herbert Boyer in 1972. What is the product? That is an antibiotic resistant gene. Actually, that is isolated from a plasmid of Salmonella typhimurium. It was linked with a plasmid vector and transferred into E. coli. As a result, the gene was expressed and multiplied in E. coli, and we get that protein, antibiotic resistant gene. It is isolated from a bacterium named Salmonella typhimurium. Then we have studied the basic steps of recombinant DNA technology. Then next is, in order to do this process, we need many tools. They are known as tools of recombinant DNA technology. We can study detail about the tools of recombinant DNA technology in the next session. Okay, students, thank you for watching this video.